Welcome back to Statistical Methods. This is Week 9, Lecture 2, where I will be introducing ANOVA. So until now, we've covered a variety of mean comparison tests that essentially allow us to test how different our observed sample mean is, as compared to each of the following: a hypothetical sample mean taken from a null population with a known mean and variance, that's the one sample z-test; a hypothetical sample mean taken from a null population with a known mean but unknown variance, a one sample t-test; another sample mean taken from the same group of participants, a dependent samples t-test; and another sample mean taken from a different group of participants, or an independent samples t-test. In all of these experiments, however, we are always talking about comparing just two means, populations, or groups. Now, what if I wanted to examine the effect of an independent variable across more than two levels? So, for example, the anchoring bias is people's tendency to base numeric judgments on earlier presented numbers, even when these numbers are randomly chosen or otherwise irrelevant to the judgment at hand. In an experiment, participants are asked to estimate what percentage of countries in the United Nations come from the continent of Africa, experimentally manipulating the anchor provided. So some participants saw a 65% anchor. So before making this judgment, participants were asked whether they thought the actual number was higher or lower than 65%. The 10% anchor, so these participants, before making their judgment, were asked whether they thought the actual number was higher or lower than 10%. And a no anchor control condition, where participants made the target estimate alone with no anchor provided. So the experimenter hypothesizes that participants in the 65% anchor group will provide higher average estimates than those in the 10% anchor group, with the no anchor group falling somewhere in between. Thus, we have a hypothesis about differences between three sample means. The methods we have learned until now, the independent samples t-test, doesn't allow for us to test such a hypothesis. For these types of research questions, we'll need to utilize a new framework known as analysis of variance. Which is also called ANOVA. As the name suggests, the logic of ANOVA rests on the comparison of different kinds of variance. So if the means from my three or more groups are very spread out, indicating that my independent variable is having some effect, this will inflate what is known as between groups variance. So we conduct an ANOVA by comparing this type of variance to the variance that is due to chance, random, or other factors, so anything other than our independent variable, the within groups variance. So in the anchoring example, if there were large differences between the average estimates in my three different experimental conditions, that is, the sample means are far apart from one another or more variable, my between groups variance will increase. And I will take this as evidence that my independent variable, so which anchor participants saw, has an effect on my dependent variable, which is their estimate. So if the anchoring manipulation doesn't have this kind of effect on percentage estimates, then I will expect low variability in my sample means. In this case, variability in my sample means will be based on the same factors as my within groups variance. So anything other than the independent variable. Now, the key statistic in the ANOVA framework, which we'll learn to actually calculate later, is known as an F ratio, a ratio of the between groups variance to the within groups variance. So F equals between groups variance divided by within groups variance. Right here. So now we'll walk through this logic in greater detail. So we'll begin working within the ANOVA framework with the simplest case, a one-way ANOVA comparing three groups where each group has an equal sample size, or n. The number way part tells us how many independent variables we're examining. It turns out that ANOVA also allows us to examine the influence of multiple independent variables and their contributions at once. And this is known as a factorial ANOVA, and we'll be covering this in the next chapter. So the null hypothesis in an ANOVA is that all of the populations being compared have the same mean. So mu1 equals mu2 equals mu3. You can see that right here between three groups. Thus, in the example I used before, the null hypothesis would be that the anchoring condition has no effect on percentage estimates. Thus, these estimates are equivalent across the three anchoring groups. The research hypothesis, then, is that there is some difference among these population means. And ANOVA tests whether your sample means differ from one another more than would be expected if there were no true difference between our three population means. Importantly, any deviation from mu1 equals mu2 equals mu3 would count in favor of the research hypothesis. For example, if mu1 is less than mu2, but mu2 is equal to mu3, that's still in favor of our hypothesis. If mu1 equals mu2, but mu2 is greater than mu3, that's still in favor of our hypothesis. And if mu1 is greater than mu2, which is greater than mu3, that also counts in favor of our hypothesis. So we got used to the notion of estimating the population variance with S squared in t-tests. And ANOVA works by comparing two different types of estimates of the population variance. And these are both based on our observed data, but are calculated in different ways. One is based on the variation within each group, so within each anchoring condition for the given example. And another is based on the variation between group means, so between those three anchoring groups. Just like in a t-test, we don't know the actual population variances, and we assume that our samples come from populations with the same variance. So that homogeneity of variance assumption still applies. One way to estimate this shared variance would be to estimate the population variance separately based on each of our samples, then average these together to get a within-group estimate of the population variance. This is an average of estimates calculated entirely from scores within each of the groups or samples. Note that because we're talking about groups with equal n, we don't need to weight these estimates like we did with S squared pooled. An important consideration is that within groups, estimate of the population variance is not affected by whether the null hypothesis is true. This estimate will come out the same regardless of whether our various populations of interest all have the same mean, if the null is true, or different means, meaning the null is false, because it focuses only on variance within each population. So if whether the null is true doesn't really affect this estimate, what does? So in other words, for what reason would people in the same group experiencing the same level of the independent variable, so shown the same anchor, have different scores on our independent variable? Well, individual differences between people in response to the same situation, so personality differences could account for within groups variance. Imperfect measurement or measurement error could account for within groups variance, or other random chance factors like people's mood on the day of the experiment, which could vary between people even within one group, or which experimenter the particular participants interacted with could influence this within groups variance. So in sum, the within group estimate of population variance will be the same regardless of whether or not the null is true, and it captures any variability within our entire group of participants that is not due to our independent variable. In other words, variability due to random chance or unknown factors that cause different people in the same circumstances to have different scores. The other way to estimate the population variance would be to do so based on the variance that exists between the different groups under consideration. Where would this variance come from? It turns out that there are two possible sources of this between groups variance, depending on whether or not the null is true. Thus, unlike for the within groups variance, this estimate will rely upon whether or not the null is true. So let's consider each of these cases in turn. So if the null is true, it means that the populations that our samples come from all have the same mean. Additionally, we still assume that these populations have the same variance and are normally distributed. Thus, our three population distributions would be identical. However, even when populations are identical, so assuming the null is true, samples taken from these populations usually won't end up with identical sample means. Why? Because of the same chance, random factors, and measurement error that we talked about in the context of within groups variance. In other words, If the null is true, then the factors that influence whether our sample means are close together or very spread out are the same factors that influence the variability among scores within each condition. So in the left panel, so this is imagining that the null is true, are 3 identical population distributions with lower variability, and 3 sample means with lower variability. And the means are down here. So you could have different sample means drawn from these 3 identical population distributions, even if these populations don't differ as a function of the independent variable. In the right panel are 3 identical population distributions with greater variability, and 3 sample means with greater variability. So even though the populations have the same mean, our sample means still can be different. Importantly, what this means is that the variable between the means of samples taken from, or the variance between the means of samples taken from identical populations is directly related to the variance of the scores in each of those populations. This means that it should be possible to estimate the variance in each population from the variation in the means of our samples. Such an estimate is called between groups estimate of the population variance. This would require working backwards from what we're used to, using the variance in our distribution of means to get the variance in our populations, as opposed to the other way around, using our sample variance to get the variance of the distribution of means, as we did with the one sample t-test. So in sum, when the null hypothesis is true, the between groups estimate of the population variance is influenced by the same chance factors that influence within groups estimate of the population variance. But what if the null is false? In other words, what if our populations of interest actually don't have the same mean? Or in other words, our independent variable is exerting some kind of effect on our dependent variable. So if the null is not true, it means our populations of interest have different means. In this situation, variability between our sample means will come from 2 sources. So one source is chance factors causing variability within the populations, just like when the null is true, and also differences in population means, or in more practical terms, because our independent variable actually has some effect. So the larger the difference between our populations, the bigger the effect of our independent variable, the larger this source of variance will be, this between source of variance. So in the left panel, the null is true, and the populations have identical means. And you still see some variability in sample means, but that's only due to within population variability. But in the right panel, the null is false, and populations have different means, so these distributions are centered around different means. And variability in sample means is due to both within population variability and this difference in population means caused by the independent variable. Notice here the means are more spread out for the samples than they are when they come from essentially the same population. Now, the difference in the amount of between groups variance that exists based on whether or not the null hypothesis is true is the key to understanding ANOVA. So if the null is true, the between group variance only reflects variation within our populations that is due to chance factors. But if the null is false, the between group variance reflects variation within our populations due to those chance factors, but also variation between populations due to some effect of our independent variable. So here is a table from the textbook to conceptualize this. So if the null is true, both the within and between subjects estimates of the population variance are based on the same thing, chance variation within our samples. Thus, we expect these estimates to be similar. So with a ratio of about 1 to 1. But if the null is false, the between subjects estimate of the population variance should be larger than the within subjects estimate, since it includes variance that is due both to the chance variation within our samples and an effect of our independent variable. so differences in population means. Thus, we expect our between subjects estimate to be larger than our within subjects estimate, with a ratio that is greater than 1 to 1. So in other words, if the null is true, between subjects or between groups variance divided by within group variance should be approximately 1. These should be about the same size. But if the null is false, between group variance divided by within group variance should be greater than 1, meaning that the between group variance is larger than within group variance. Now, this ratio of between group variance to within group variance is known as the F ratio, and F serves as the central test statistic of interest in ANOVA. Now, because this number is based on variance, which is always positive, it can only be positive, unlike t-scores or z-scores. This statistic follows an F distribution, another hypothetical mathematically derived distribution with known properties like the t-distribution. And this distribution is what allows us to do hypothesis tests with F, so about differences between multiple sample means, answering the question, how extreme or large does my F ratio have to be to reject the null hypothesis that there is no difference amongst my population means? The precise shape of the F distribution will depend upon two types of degrees of freedom, and we'll talk more about this later, but will span from 0 upwards and generally look something like this. It spans from 0 upwards because F is always positive. Yeah, it generally has this fairly strong positive skew. So you might rightly be asking at this point, is it actually useful to test whether there is any difference at all between the different group means? In other words, won't we usually be more interested in which groups differ and by how much? In short, the answer is yes, but we'll discuss how this is handled and why within ANOVA in future lectures. Some useful resources for understanding ANOVA logic can be found here. And I'll leave you with a class problem. In what types of contexts would you use a one-way ANOVA? If the null is true, what would you expect the F ratio to be? If the null is false, what would you expect the null ratio to be? And what are the two sources of variance that contribute to between groups estimate of the variance? So pause it here and then record answers to these questions and then play the video. So in what types of contexts would you use a one-way ANOVA? Well, if your experimental design has more than two groups. So for example, if you have one independent variable with 3 levels, or if you have 2 independent variables with 2 levels. If the null is true, what would you expect the F ratio to be, roughly? So that would be approximately 1. So within groups variance and between groups variance should be almost the same. But if the null is false, what would you expect the F ratio to be? Well, greater than 1. You would expect your between groups variance to be larger than your within groups variance. And what are the two sources of variance that contribute to the between groups estimate of the variance? Well, one is chance variation, but the other is variation due to the effect of the independent variable. That's all for today. See you next week.